Pythium habit and habitat. The genus is represented by about 92 species which are cosmopolitan in distribution. The species may be aquatic, terrestrial or amphibious in habitat. The fungus is facultative parasite and lives saprophytically on the moist humus in soil and attacks seedlings at the soil level. Thereafter, it lives as parasite. It also grows readily on floating vegetable matter and decaying animals. Common diseases caused by Pythium, damping off, soft rot, wheat rot, or the foot rot of the seedlings are common diseases caused by the fungus. P. Debayanum causes damping off disease of tobacco, mustard, chilies and cress seedlings. Excessive moisture, presence of too much decaying matter in the soil, overcrowding of the seedlings and insufficient exposure to air and light are the predisposing causes for this disease to appear. Symptoms of diseases caused by Pythium. Young seedlings are affected near the ground level. Initially no external symptoms of its presence are noticeable on the host, but after it has gained a firm hold, the seedlings become pale green and show a weak, shriveled place of girdle of brown color on the stem near the surface of the soil. At this point the cortical cells are killed, making the tissue weak. The infected tissues appear soft and water-soaked. Infected seedlings collapse. The upper part of seedling bends or topples over in a characteristic manner. This peculiar symptom of disease is known as damping off. The disease occurs in two stages. 1. Pre-emergence phase. Young seedlings are killed before they come out of the soil. 2. Post-emergence phase. Toppling over the infected seedlings after they come out of the soil surface. Vegetative structure. The mycelium appears as white, fluffy mass. It is well developed, branched, inter- or intracellular, multinucleate and without septa, coenocytic. Haustoria are not produced. Food material is absorbed. Hyphal walls. Septa appear only in association with the sex organs formation. Cell wall lacks chitin and instead it is composed of glucan and cellulose. Cell wall encloses vacuolated cytoplasm, mitochondria, a nuclei, endoplasmic reticulum, ribosomes, oil globules and glycogen in the form of reserve food material. The branched hyphae are very much similar to the filaments of Vaucaria. Reproduction. The fungus reproduces by asexual and sexual methods. Asexual reproduction. Asexual reproduction takes place by the formation of sporangia. Sporangia may be terminal or intercalary in position and are produced on the aerial hyphae. There are no specialized sporangiophores to produce sporangia. The terminal or intercalary position of a hypha enlarges and becomes spherical and starts off function as sporangial initial. It gets cut off from rest of the mycelium by a transverse septum. Sporangia are multinucleate and may be filamentous, tubular or globes in shape. Sporangia germinate indirectly or directly. Indirect germination. Under favorable conditions, high humidity, sporangia behave as zoosporangia and produce zoospores. The zoospores are reniform, uninucleate and biflagellate. One flagellum is of tinsel type and the other is of whiplash type. Germ tube develops from the encysted zoospore which infects the host by entering through the stomata or by directly penetrating the epidermal cells of the host. Direct germination. Under unfavorable conditions, dry conditions, the sporangia behave as conidia and are called conidiosporangia. It does not produce zoospores but germinates directly to form a germ tube. Sexual reproduction. Sexual reproduction is oogamous and occurs under unfavorable conditions at the end of the growing season of the fungus. Pythium is homothoric, i.e., male and female sex organs develop in close proximity of each other either on the same or different hypha embedded in the host tissue. However, some species are heterothoric. Male and female sex organs are called antheridia and oogonia, respectively. Antheridium. Each antheridium is club-shaped, paragynous present to the side of the archegonium, and multinucleate. It arises as branch of the stalk of the oogonium, monoclonous, or on separate hypha, diclinous. Male branch gets slightly inflated at the tips. A transverse septum appears between the inflated portion and rest of the hypha. The inflated portion is called antheridium. On maturity all antheridial nuclei except one degenerate and the surviving nucleus is clade functional nucleus. Many antheridia 1 to 6 are sometimes seen in association with one oogonium. Oogonium, it is a globose, spherical, smooth-walled structure. It develops at the tip of the hypha terminal. Sometimes it is also intercalary in position. The tip of the hypha swells up and it soon cuts off from the main hypha by the formation of a septum. On maturity, its protoplasm is differentiated into an outer thin spongy portion lining the eugonial wall, the periplasm, and the inner, granular denser rounded portion, the euplasm or oosphere. The euplasm contains a single female nucleus, while the other nuclei within the periplasm degenerate. Fertilization. As Antheridium matures, it bends towards the eugonium gaintangial contact to which it applies itself closely. The wall of the antheridium and eugonium dissolves at the point of contact. Antheridium developed a fine fertilization tube or penetration tube which penetrates the periplasm and reaches the euplasm. The functional male nucleus enters the eugonium through fertilization tube and fuses with the female nucleus and forms a diploid zygotic nucleus. Oospore. The zygote nucleus secretes a thick double wall and called oospore. The outer wall is thick and is called exene, while the inner wall is thin and is called intene. The oospore is a resting spore. It is filled with plenty of reserve food material. Germination of oospore. Oospore germinates in the presence of moisture. The exene ruptures and intene comes out in the form of germ tube. The zygotic nucleus divides. The first division is meiosis. If the temperature is high 28 degrees Celsius, the germ tube directly develops into mycelium. However, if the temperature is low 10–17 degrees Celsius and enough moisture is available, the contents of the oospore migrate into the sporangium through the germ tube. The sporangium behaves like a zoosporangium and produces zoospores as in asexual reproduction. Control measures. Proper drainage system of the field should be maintained. Waterlogging in the soil and excessive watering should be avoided. Adequate ventilation should be provided. Too thick sowing on the seed beds should be avoided. Soil should be sterilized by using steam, dry heat, or chemicals such as formaldehyde diluted in water, 150, and Bordeaux mixture, ½ gallon per square foot. Soil can be fumigated with chloropicrin or methyl bromide. Detrenching soil with 0.2% phytolin, 0.5% perinox and 0.2% flit is very effective method to control the damping of disease. For pre-emergence phase of disease, the seeds should be treated with BILTOX50 and Arasan. Chemicals like chlorineb can be used in seedlings of pepper, bean, tomato, etc.